All right, good morning, everyone. I believe we left off right here. Someone said good morning. Wow, amazing. So we were talking about the importance of maintaining a sterile technique when you're working with microorganisms. So if you're collecting a sample from a patient or if you're working in Microbiology 210 lab, you need to make sure that you're not contaminating it with stuff from your hair, your skin, whatever, the dirt from the floor. You want to make sure that what you are working with is really the thing that you think you're working with. So actually, I can give you a good example. Some students were doing research in my laboratory this week, and they were growing up E. coli. And I was like, well, why don't you just look at it under a microscope and make sure it actually looks like E. coli? Turns out it wasn't E. coli. They had contaminated it with something. And so the experiment they thought they were doing is not the experiment they were doing at all. So it really does matter. It can blow your whole research project out of the water if you're a research student. It could kill someone if you're working in a hospital. So making sure that you have good, proper sterile technique really is important. So when we talk about this in lab, make sure that you're following the procedures that we teach you. All right, one of the things that you'll learn in lab is the streak plate method. So you usually put a small amount of bacteria up at the top part of a Petri dish, and then you take your loop and you streak through it and bring the bacteria out, and then you streak through it again and bring the bacteria out. And the purpose of this is you're basically diluting the bacteria. You're making it so that you can get individual colonies of bacteria and you can actually see what you're working with. Up here, you can see there's so many bacteria that it's hard to even tell what's going on. But as you streak down and then streak out, you start to get nice individual colonies, and you can clearly tell that there is more than one type of bacteria growing on this plate. Now, if this was a patient sample, depending on where it came from, you might expect that there was more than one bacteria. If it came from their skin, there's tons of bacteria on your skin. But if it was a blood sample, well, your blood's usually sterile. So unless you have an infection, you wouldn't expect there to be anything there. And so if you've got 5 types of bacteria growing on a blood sample, you probably screwed something up. Maybe there would be one type of bacteria if there was an infection, but I doubt there would be 5. All right. You need to know a little bit of chemistry. You've all taken at least one chemistry class before you've taken this class. You haven't taken, well, has anyone taken organic chemistry yet? Okay, like 2, maybe 3. You do need to understand a little bit about organic chemistry. So there are inorganic chemicals, salts like sodium chloride, calcium carbonate. Carbon dioxide, even though it has a carbon atom in it, is considered an inorganic molecule. If you burn something, you make carbon dioxide. It has nothing to do with life. There are also organic chemicals. So these are carbon-containing molecules, things like DNA, proteins. And the reason that carbon is such a good molecule for life is that you can make lots of different shapes out of it. You can make straight chains, you can make chains with branches, you can make rings, and so it gives a lot of diversity to life. We can also add things to these carbon chains, which we call functional groups. So adding an oxygen and a hydrogen, this is a hydroxyl or alcohol. So ethanol is 2 carbons, an oxygen, and a hydrogen. A carboxyl group is a double bonded oxygen and an oxygen and a hydrogen on that carbon. So why does this matter? Well, when we talk about archaea and bacteria, the lipids in their cell membranes look different. The archaea have this and the bacteria have this. So in order for you to understand the differences between archaea and bacteria, you have to know the difference between a hydroxyl and a carboxyl. So make sure that you know these two functional groups. Other things that are important. Amino acids have amine groups, which is a nitrogen and two hydrogens. Phosphate groups, these are found in DNA and RNA. So these are important molecules for understanding what we're even talking about for the next couple of weeks. All right, so I'm going to test your chemistry and see if you remember what you should have learned in 160 or whichever chemistry class you took. All right, so a solution that has a pH of 3, is that neutral, acidic, basic, or alkaline? All right. Okay, so that is acidic. Good. Your chemistry professor will be very happy to know that you all remember that. So these are the molecules that we talk about when we talk about living organisms. We have proteins, we have nucleic acids, we have lipids in the cell membrane. All of these are made using carbon. Carbohydrates. So you usually think about these when you look at the nutritional information on the back of your Pop-Tarts or whatever. How many carbs are in it? Lipids and proteins. And nucleic acids. So why do we need all these things? Well, they give structure to our cells. They can be messenger molecules. We can use them to store energy. Proteins are used as enzymes to speed up chemical reactions. DNA is the source of our genetic information. So if we look at carbohydrates, we have a monosaccharide, which is a single sugar. We have a disaccharide, just two sugars. Or you can have a polysaccharide, which is many sugar molecules linked together. And a polysaccharide, cellulose is an example. So cell walls of plants are made out of cellulose. So, well, why do we care about that? If you're a cow, you care a lot about cellulose. Even if you're a human, you eat vegetables and fruits and things, there's cellulose there. Only prokaryotic organisms can break down, or single cell organisms can break down cellulose. So if you didn't have bacteria living in your intestines and you ate a carrot, you might as well eat a rock. It would do absolutely nothing for you if you didn't have the bacteria living in your intestines to help you break down and get some of the nutrients out of that food. So only bacteria, fungi, and protozoa are capable of digesting cellulose. So a termites eat wood, right? That's what they live off of. But the only way they get any food out of that wood is because of the bacteria living in the intestines of the termite. It's the bacteria that actually extracts the nutrients out of the wood for the termite. And then the termite's able to absorb them. We also use agar when we are making media. So if you're making a petri dish, you add agar to it to create this kind of semi-solid stuff that you can grow bacteria on. All right, lipids. So remember I said you needed to know some of those functional groups. So that was a carboxyl group. So lipids are part of cell membranes. They're also really good for energy storage. All right, amino acids. So all of your proteins are made out of amino acids. Notice that we have lots of different kinds of functional groups here on amino acids. I'm not going to expect you to memorize them all, but you should definitely recognize the carboxyl group and the amine, hence the name amino acid. All right, when we talk about proteins, we refer to the structure of the protein in 4 different levels. We have the primary structure, which is the amino acid sequence. So that's just glycine, asparagine, tryptophan, glutamine. So that is the primary structure. The secondary structure is things like alpha helices and beta sheets. So this is what they look like. The structure will loop around itself, creating a helix. The tertiary structure is the 3D structure. So you can actually have a beta sheet and an alpha helix kind of all combined into this one big blob. And so we refer to that as the tertiary structure. And the quaternary structure is if you have more than one piece. So we've got a purple piece of protein and an orange piece and a green piece. So these are actually different pieces. They're made separately, but then they all come together in the end to form one large protein. So you need to know these 4 different levels of folding or complexity within proteins. All right, so why do we care about the shape of a protein? Well, the shape determines its function. So in order for an enzyme to work, it has to have a specific shape. So if we have an enzyme that's going to break down a carbohydrate, let's say glucose, and use it as an energy source, it has to have a specific shape so that the glucose molecule fits in the protein and it can perform its function. Things like antibodies, which help our immune system, they work because they have a very specific shape that matches the shape of the virus or bacteria that they are attacking. So when we talk about proteins, their native state is their natural tertiary or quaternary structure. So if you were to look inside your cells and look at hemoglobin, it has a specific shape, size. The denatured state is when we mess that up. So if you cook an egg, you are denaturing the proteins. What happens when you cook it? It goes from this slimy stuff into this nice, hard, jello-looking stuff. I don't know. So when you change or denature a protein, you totally alter what it does. So the function of the protein in egg whites is to provide nutrients to the developing chicken. But when you cook it, it turns into this stuff that's no longer useful to the chicken anymore. Enzymes in your own body, if you heat them up, if you add harsh chemicals to them like bleach, you damage the proteins and they no longer function. Their shape and size changes, and so they can't work anymore. So in the next unit, when we talk about antibiotic drugs or chemicals like bleach that we use to disinfect things, the reason most of those chemicals kill bacteria is because they denature proteins in the bacteria. The bacteria can no longer function and they die. All right, nucleic acids. We won't go into the structure of this too much. We'll talk about it more when we go into the genetics of bacteria. Hopefully you all know, in general, what DNA looks like, though. All right. So why are phospholipids important as biological molecules? One of my daughters would probably answer A. She likes to eat butter, like straight out of the tub. That's creepy. Okay, good. They form cell membranes. So if you don't know what a phospholipid is, then go back and read through chapter 2. Okay, any questions about the molecules we talked about before we move on? All right. Before we get into lecture 3, there's a couple of things I want to mention. If you go on to Blackboard, I put a couple of new folders and kind of rearranged the organization of things so that hopefully it's a little easier to find what you're looking for. So there are the PowerPoint lectures. The audio recording that I make of each lecture is now in its own folder. All of your LearnSmart assignments and quizzes are in their own folder. So instead of having to scroll down and find what you're looking for in this one folder, I've kind of separated them out into different folders. And your lab assignments are all in one folder. All right, so for the rest of the lecture, we're going to talk about what are prokaryotic cells, what do they look like, what are their features, what makes them different from eukaryotic cells. Before we do that, though, if you have read through your syllabus, you'll notice that there was a little thing in your syllabus about extra credit points. If you do community service, I'm willing to give you 2 extra credit points for every 1 hour of community service that you do. So if you're planning on going to nursing school or many healthcare related fields, they want to see that you've been involved in community service. If you're going to be a nurse, what you're doing is serving the population. So it demonstrates that you actually are interested in helping people around you. So there is an opportunity coming up in just over a week. It's the Public Affairs Week, September 9th through the 15th. And there are several service opportunities in there. On Saturday, September 9th, there is Into the Streets, which is a student volunteer opportunity. They're going to go around and help clean up some neighborhoods. There is also, which day is it on? On Monday, September 11th, there is the Hunger Banquet. So this isn't necessarily a service opportunity, but what you do is you go to the PSU and they basically randomly assign you to a group. And the amount of food you get to eat for dinner that night is based on your social status. So they may assign you to be in a third world country group, in which case you get a little teeny bowl of soup to eat for dinner that night, and that's it. Or they may assign you to be in the 1% group, and you get to have lots of food to eat for dinner that night. And then they have a discussion, and they talk about why is this? Why do we think it's okay for some people in the world to have a little cup of soup and other people to have a massive banquet? So, not necessarily community service, but I definitely think it's an eye-opening experience and a good opportunity to learn. So I'll give you extra credit for that as well. There's also a voter registration campaign, so if you participate in that. In order to get credit, I have posted a, not a sign-up sheet, but a sheet that allows you to get a signature from whoever is in charge of the event to demonstrate that you actually attended the event. So that is posted on Blackboard next to the syllabus. So if you go on Blackboard, under Getting Started, just below the syllabus, there is a log sheet that you can bring to whatever event and have the person in charge sign it so that you can get credit. If you're involved in a fraternity or sorority and you have a service project, that's cool too. It doesn't necessarily have to be something that I bring up. If you're volunteering at Cox right now, then take it in and get your sheet signed so that you can get extra credit. Any questions about that? Yes?
Is that due at a certain time, or is that
Yep.
throughout the semester?
Throughout the semester. Yep. Yes?
Is it unlimited? How many?
So you can do a maximum of 5 hours, so 10 extra credit points. Okay? I mean, you can do more than 5 hours, but you only get 10 extra credit. All right, so this is what I hope that you will learn from this lecture. You should know the structures that are common to all bacteria and some structures that are unique. So all bacteria, or virtually all bacteria, have a cell wall. But only some bacteria have a flagella. So you need to recognize what is common to all or most bacteria and what is unique to some bacteria. You should be able to describe the shapes and clustering of bacteria, the function of different appendages. You should know how chemotaxis works. Chemotaxis is the ability of a bacteria to swim to where it wants to go. So if it senses a food molecule, it'll swim to that food molecule. So that is chemotaxis. And you should know the differences between archaea and bacteria. All right. Overall, the differences between bacteria, archaea, and eukaryotes. The way they package their DNA is where the names come from. Eukaryotic cells have a nucleus. Prokaryotic cells don't have a nucleus. Their cell wall makeup. Bacteria's cell walls are made out of peptidoglycan, and archaea cell walls are made out of either protein or pseudomurine. We'll talk about that in a little bit. Internal structures. Bacteria and archaea have no membrane-bound organelles, whereas eukaryotic cells do. All right, so let's look at our bacteria here. In all bacteria, they all have a cell membrane. Every living organism has to have a cell membrane. They have to have genetic material. So they all have DNA. It's just not in a nucleus. They have ribosomes. You have to be able to translate RNA into protein, so you have to have ribosomes. And cytoplasm. So this is the watery substance inside the cell. Now, there's a lot of things that are in some bacteria, and we will talk about these as we go through the lecture. So you definitely need to know everything that's under the all bacteria listed here. Bacterial size. So bacteria can be very small. Most bacteria are about 1 micrometer, which is too small to see without a microscope. However, some bacteria, like Thiomargarita namibiensis, can be as large as 750 microns, or about 0.75 millimeters. You can see a millimeter, like without a microscope. This guy's almost a millimeter big. You can see it without a microscope. In fact, it's bigger than the cells in your own body. It's a very large bacteria. We also have mycoplasma, which is less than a micrometer. So you almost need an electron microscope to be able to see these bacteria. And nanobacteria, they can even be smaller than that. When we talk about bacteria, in addition to their size, we also define a lot of the characteristics of bacteria based on their shape. Is it long and skinny? Is it round? So there are three main shapes. Coccus, which is a sphere or maybe a slightly oval shape. You have bacillus, which is tube-shaped. And you have vibrio, which is curved or half-moon shaped. Now, bacteria aren't perfectly shaped. So for a coccus, this is a coccus, but this kind of teardrop-shaped thing is also considered a coccus. It's mostly round. For bacillus, this kind of long stick-shaped one is a bacillus as well as this one. And for vibrio, we can have, a nice half-moon shaped curve or kind of a bent, slightly curved. So we as scientists like to give names to things, but as you continue on in your scientific career, you'll see that not everything falls exactly under one category. Life is very diverse. All right, there are other types of bacteria which don't fall into those main three categories. We have Spirillum and spirochetes that have kind of a corkscrew shape to them. We also have branching filaments. So these almost look like a fungus, but they're actually a bacteria. In addition to the shape of the bacteria, we can also have different clustering of bacteria. So you can have a diplococcus, which is two cocci, but they stick to each other. And so when you look at them under the microscope, they're usually in pairs. You can have tetrads, so four. Staphylococci, we frequently refer to it as grape clusters, so it's just, there's no structure like with tetrads, it's just a big blob of cocci. Streptococci look like beads on a string. It looks like a string of pearls. And then Sarcina are large cubes. So you may have 8 or 16 bacteria in a very geometrical structure. All right, bacilli can have similar structures. You can have a diplobacilli. You can have a streptobacilli. Spirilia, on the other hand, usually are just a single cell or maybe in short chains. All right, so what are the three most common shapes for bacteria? Good. So A was the correct answer. So Bacillus, coccus, and vibrio. All right. For the bacterial cell wall, the function is to protect the bacteria from the environment. If you don't like your environment, you can change it. You can go to the thermostat, you can put on a jacket. There's a lot of things you can do to change your environment if you don't like it. If a bacteria is in an environment it doesn't like, it doesn't necessarily have the ability to change it. So if it starts getting cold, if the pond that the bacteria is living in freezes, bacteria can't change that, it just has to deal with it. And so the cell wall helps the bacteria to deal with inhospitable conditions. One of those is osmotic pressure. So if water molecules start moving into the cell will expand. And if it didn't have a cell wall, it would pop like a balloon. So the cell wall helps to keep the bacteria intact. The main molecule in bacteria for the cell wall is peptidoglycan. Another name is murine. So this is a synonym for peptidoglycan. And what it is just long chains of sugar molecules with short amino acids linking the two together. On the other hand, archaea has a molecule called pseudomurine. So murine in bacteria, pseudomurine in archaea. But even though most bacteria have peptidoglycan in their cell walls aren't identical. We have gram-positive bacteria, we have gram-negative bacteria. So for gram-positive bacteria, there's a very thick layer of peptidoglycan on the outside of the bacteria. For gram-negative bacteria, there's a thin layer of peptidoglycan sandwiched between two membranes. There's the cell membrane and an outer membrane. So if I were to show you a picture of one of these on your exam, you should be able to tell me whether it's gram-positive or gram-negative. Now this is one of the main staining techniques that you will use in microbiology lab, is being able to stain for gram-positive or gram-negative bacteria. So the purple bacteria on this slide are your gram-positives, and the more reddish colored are the gram-negatives. On the gram-negative bacteria, the outer membrane also has something called lipopolysaccharide. So that's the little chains coming off of the outer membrane here. Those give some protection to the bacteria. From an immune system standpoint, they actually tell the immune system that this is a foreign invader. So if you get a gram-negative bacterial infection, your immune system can detect the lipopolysaccharide and know that this bacteria is not supposed to be there. All right, on the outside of the cell wall, we have certain structures. We have flagella. So the flagella is used for movement. The flagella has a long filament, a hook, and a basal body. So the basal body actually works just like a motor. It spins and it causes the filament to turn like a propeller blade. So it's literally like a boat propeller. We also have fimbriae and pili. These provide attachment. So if you think about a bacteria, if it's in your intestines, what happens to the contents of your intestines? Do they stay there? No. Hopefully at least once a day you use the restroom. So if you're a bacteria and you want to stay in the intestines, you have to have a way to attach yourself so that you don't get swept away. And so pili and fimbriae serve that purpose of allowing bacteria to attach to a surface in order so that they can stay put. Same thing if you live in a lake. Let's say you're a bacteria and you want to attach yourself to a rock so that you don't get swept away and you can stay right there. Your pili or fimbriae would allow you to do that. All right, for flagella, there are different arrangements of flagella. We have monotrichous, so that's one flagella coming right out of the back of the bacteria. We have lophotrichous, which means that we have several flagella, but they're all coming out of the back. We have amphitrichous, which means we've got a flagella on either side. And finally, peritrichous, where we have flagella totally surrounding the bacteria. So again, if I were to show you one of these pictures, you should be able to tell me which arrangement it is. All right, so bacteria have flagella so that they can move where they want to go. And we call this chemotaxis, or phototaxis if it's a photosynthetic bacteria. So if you are a photosynthetic bacteria and you live in a pond, where do you want to be? You want to be at the bottom? You want to be at the top? But if you're photosynthetic, sun's coming from above, so you want to be near the top. So phototaxis would be the movement toward light. So in this test shown here at the bottom, you have a beaker full of, let's just say it's water, and you put a test tube with water into the beaker. Well, if you have water and water, there's nothing to attract the bacteria. And so you'll get a few bacteria going into the tube with water, but that's just random. They're just swimming around. It's kind of like cruising downtown on Saturday night. You just kind of end up someplace, whether you meant to or not. C, here the green liquid is a sugar molecule, something that the bacteria actually wants. And so you can see that there are many bacteria which have gone up into the tube here. They are intentionally going toward this source of potential food. We can also have a repellent, so let's say we add a little bit of bleach into this water, and so the bacteria are now all swimming away from it. They don't want to be there. There's two directions that the flagella can rotate, clockwise or counterclockwise. Counterclockwise means that the bacteria is moving forward or running. Clockwise causes the bacteria to actually start flipping and spinning, and that's how the bacteria changes direction. So if we do counterclockwise, we go in a straight line, and then if we do clockwise, the bacteria starts flipping, and it can then change direction and go somewhere else. So this is what that would kind of look like. So T stands for tumble, R stands for run. So if we have a bacteria and there's nothing there, this is just water, there's no attractant to tell the bacteria where to go, the bacteria will run, tumble, and it'll just keep doing this, and really it's just wandering around. It doesn't really go anywhere. But if we actually have a chemoattractant, something the bacteria is interested in, it doesn't just go in a straight line. The bacteria can't actually see. So it can't see, oh, the food's over there. All right, I'm going to go over there. It just knows that it's starting to sense the chemical. And so it might actually start going in this direction. And it gets this far and it says, wait, I don't sense the chemical anymore. And so it kind of turns around and it goes back. And then it starts going this way and it says, oh, I sense the chemical. Wait, I'm getting stronger. Ooh, closer, closer. So it's kind of like hot cold, the game used to play as a kid. So you can see that the bacteria, even though it's heading toward this chemical, it doesn't just go in a straight shot. It goes a little bit, it turns, and it kind of readjusts itself. All right, so watch a quick video about this.
When Echinacea coli moves to a medium that lacks a concentration gradient, the cell travels, stops, or tumbles, and then continues moving in a new random direction. When the flagella rotate counterclockwise, the flagella form a tight bundle and propel the cell forward in a run. After a brief period, the direction of rotation is reversed, causing a tumble. As cells move up a chemical gradient, the runs are longer than when they travel down the gradient. The overall result is random movement in the absence of a chemical gradient and movement toward a chemical when a gradient exists. Attractant chemicals bind to chemoreceptors called methyl-accepting chemotaxis proteins located in the cell membrane. The cytoplasmic side of the MCP interacts with two proteins, key W and key A. When the MCP is not bound to an attractant, it stimulates key A to phosphorylate itself using ATP. Key A autophosphorylation is inhibited when the attractant is bound to its MCP. Phosphorylated key A can donate its phosphate to the response protein, key Y. When key Y is phosphorylated by key A, it moves to the flagellum. This causes the flagellum to rotate clockwise and tumble. Thus, a decrease in attractant promotes short runs and tumbling. Key W is an adapter protein, mediating communication between receptors and key A. The phosphate on key Y is continually removed by key Z. The short lifetime of phosphorylated key Y means that the bacterium is very responsive to changes in attractant concentration. When no attractant is present, the cell maintains intermediate levels of key A phosphate and key Y phosphate. This produces a normal run-tumble swimming pattern. When a chemoattractant binds to MCP, the levels of key Y phosphate drop. The lack of phosphorylated key Y results in counterclockwise rotation of the flagella and long, smooth runs. Escherichia coli must be able to ignore past stimulus responses so that it can compare the most recent attractant concentration to the immediately previous one. This is accomplished by methylation of the methyl-accepting protein. Key R continuously methylates MCP, whereas phosphorylated key B removes these methyl groups. When the attractant is bound to MCP, the complex becomes a good substrate for key R and a poor substrate for key BP. The levels of key YP and key BP drop because phosphorylation of key A is inhibited. Removal of the attractant causes the overmethylated MCP to stimulate key A autophosphorylation, and the levels of key YP and key BP increase. This simultaneously induces tumbling and demethylation, and the system returns to a lower level of key A autophosphorylation.
All right, now aren't you glad I'm not going to make you memorize that? Yes. Yeah. Okay, so the point is, it's actually really complicated. The bacteria has evolved many ways to be able to regulate which direction it's going, how fast it tumbles. So you don't have to memorize any of those molecules. All you need to know is that the bacteria is able to regulate. It senses a signal, it responds to that signal, and it changes its ability to run or tumble. I'm actually surprised your textbook even includes that as a, like that would be like a graduate school video for understanding how that works. All right. There are certain bacteria, the spirochetes, which have a very unique flagella. So instead of the flagella being on the outside of the cell and working as a propeller, the flagella is actually in between the cell wall and the cell membrane. And so instead of whipping around like a propeller, it actually causes the corkscrew-shaped bacteria to turn like a drill bit. So the entire bacteria starts twisting and turning just like a drill bit would. And that allows the bacteria to kind of drill its way through whatever it happens to be in. All right, fimbriae. So we talked about how pili and fimbriae are important for attachment. So you can see that it looks like this bacteria has been infected by a cactus. They really are just completely covered with these fimbriae, and you can kind of think of it as Velcro. It will stick to whatever it comes into contact with. All right, pilus. So there's a specific type of pilus called a sex pilus, which allows the bacteria to exchange DNA from one bacteria to another. It's generally found in gram-negative bacteria, and the process of sharing DNA from one bacteria to another is called conjugation. So why is this important? A bacteria that has antibiotic resistance will frequently have that antibiotic resistance on a small piece of DNA called a plasmid. And it can share that small piece of DNA through this sex pilus. So now you can have antibiotic-resistant E. coli that shares its antibiotic resistance gene with Shigella. And Shigella is what causes dysentery. So now instead of just having antibiotic-resistant E. coli, you have antibiotic-resistant dysentery. And so this is the problem with antibiotic resistance. Every time we use antibiotics improperly, not only are we making antibiotic-resistant E. coli, but we could be making other organisms antibiotic-resistant because they share some of that DNA. All right, surface coatings. So some bacteria on the outside of the cell wall have additional protective coatings. The S layer, these are frequently found in archaea because of the nasty environments that they live in. So if you think about the hot springs at like Yellowstone National Park, in order for a bacteria to live in boiling water, it has to have a way to protect itself. And it protects itself with a shell of protein called an S layer. We also have the glycocalyx, which can be a slime layer or a capsule. So if you look at this slide down here, the bacteria is the little thing in the center. The white area around the bacteria is the capsule. So the bacteria can not only increase its size dramatically by coating itself, but this capsule also prevents the bacteria from drying out. It can prevent white blood cells from being able to phagocytose and destroy the bacteria. All right, we'll go ahead and stop there, and then we'll talk about biofilms next week, or on Thursday.